various institutes, including UCL in London and the University of Liverpool. And in 1999, he took up a chair in cardiac physiology at the University of Manchester in the UK. Following on from that, the following year, he got a BHF professorship. And since then, he's been working on calcium in the heart and looking at how calcium is controlled in the heart and how this can lead to arrhythmogenesis and cardiac disease. So it's with great pleasure to introduce the floor, the virtual floor, and pass it over to David to listen to his talk. Thank you, David.
Pleasure. So you can see my screen, can you?
Yeah, it's perfect.
Okay. So first of all, Dan, thanks for the kind introduction. Thank you all for the invitation. And I hope that normality resumes sooner rather than later. So I want to talk about control of calcium in the heart. Most of it is going to be talking about free calcium, but I'm also going to have a few words to say about total calcium and calcium buffering. And really, much of what I'm going to talk about comes down to the fact that the heart has to recycle calcium, and many of the somewhat surprising findings come down to this. I'll explain this as we go along. So I'm sure everybody knows calcium in the heart started in 1883 with Sidney Ringer, who showed that calcium was required for the heart to beat. And indeed, I don't need to tell members of the European Calcium Society that calcium controls virtually every function in our bodies. More than 130 years later, we know, of course, that the reason that external calcium is required is that changes of intracellular calcium regulate function, in this case, in the heart. So this is a ventricular myocyte loaded with flow 3. Each time we electrically stimulate it, it contracts as it would in the heart. And you can see that that contraction is associated with an uniform increase of calcium throughout the cytoplasm. It's not for today's talk, but you can see that nuclear calcium and the two nuclei in the cell lag behind. We can quantify calcium. At rest or in diastole, calcium starts off at about 100 nanomolar, 0.1 micromolar, and rises to micromolar during systole. And it's this regulation of calcium in both rest and contraction that I'm going to consider today. So I'm going to give an introduction designed for those of you who don't work on calcium signaling in the heart. I'll have a few words to say about calcium buffering. I'll then talk mainly about how calcium goes up in the heart and then how it comes down, and I'll end up coming back to buffering. Okay, slight embarrassment, since I'm sure you know all this. What calcium actually does in the heart, like everywhere else, is that you have an arbitrary protein like this. This is an imaginary one, which has a certain shape. Calcium comes along and can bind to it. And because of the charge of calcium, this makes the protein change its shape. If it's an enzyme, it can change its enzymatic activity. If it's a protein such as troponin, its change of shape makes muscle contraction occur. So calcium binds. Obviously, to reverse this, calcium has to be removed, taken off, so that the protein can regain its original shape. And this is, I think, where the interesting thing about calcium comes from. If we were talking about control by phosphorylation, we talk about a phosphatase coming along and essentially destroying the link. With calcium, the amount of calcium you have in the cell or in the body is essentially invariant. So you can't destroy calcium. Calcium has to be removed from the cytoplasm, and the way it's removed from the cytoplasm is by being pumped across membranes, either into the store, the sarcoplasmic reticulum, or out of a cell on the surface membrane. And that's what I mean by the fact that calcium has to be recycled on each beat in the heart. So we record from a cardiac myocyte. Calcium in systole has to be high enough to activate the contractile proteins such that the heart can develop pressure and pump blood out. In diastole, it has to be low enough that the myofilaments are relaxed such that the heart can refill with blood. If we have this is where this all happens. We have a cardiac myocyte here. Here's the surface membrane. These are thick, large diameter cells. So they have these invaginations of the surface membrane, the transverse tubules. And many of the surface membrane L-type calcium channels are located on the transverse tubules. And calcium coming into the cell through the L-type calcium channel is one source of calcium for contraction. But it's not the only source of calcium, and indeed it's not the major source of calcium. The major source of calcium is calcium stored in the sarcoplasmic reticulum, or SR. Calcium is released from the sarcoplasmic reticulum through the specialized release channel, the ryanodine receptor. For today's talk, I have to remind you about three facets of the ryanodine receptor. The first, its open probability, PO as we call it, is increased by an increase of calcium in the cytoplasm. Secondly, PO is also increased by an increase of calcium inside the SR. And finally, we can use the drug or tool caffeine to increase the open probability PO even without any change of calcium. So during the normal cardiac cycle, calcium enters from outside into this very narrow space here. The resulting rise of calcium, because of this property, makes the ryanodine receptor open, releasing calcium into the cytoplasm and producing the rise of calcium. This rise of calcium binds to troponin, and we get sliding of the myofilaments, cardiac contraction, and the development of pressure. But if the heart's going to work as a pump, not only must it contract, it must also relax. And relaxation requires that calcium be lowered back to resting levels. And there are two ways in which calcium is lowered. One, it's taken back into the SR by the SR calcium ATPase circuit. As I'm sure you all know, circuit's activity is controlled by two things. One is the availability of calcium, and the other is this inhibitory protein phospholamban. When phospholamban is phosphorylated, as occurs during beta-adrenergic stimulation, for example, this inhibition is relieved and circuit pumps more quickly. The other way that calcium is removed is by being pumped out of the cell on the sodium-calcium exchange, which uses the energy provided by 3 sodium ions to pump 1 calcium ion out of a cell. And the stoichiometry means that each time a calcium ion is pumped out, 1 net positive charge enters the cell. There is also a plasma membrane calcium ATPase, and its exact role in the heart is still controversial. In the flux calculations I'm going to show you, we've corrected for that. So we can measure both calcium influx and calcium efflux using the patch clamp because they both are associated with membrane currents. The other thing I want to emphasize is that the events I've described happen each time the heart beats. And because the heart is in a steady state, on each beat, the calcium influx must equal the efflux. That's the definition of a steady state. So the amount of calcium that enters here is equal to the amount of calcium pumped out here. The amount of calcium released here is equal to the amount of calcium taken up here. That happens on every beat. And I think that contrasts with some of the systems that some of you work on, particularly the non-excitable cells, where calcium regulation occurs over much longer periods of time. In the heart, in a typical human, these events are back in balance within less than a second. I can show you that the heart is in calcium flux balance and use this to demonstrate the experimental approach we take. So these experiments are done under patch clamp. So we use a feedback amplifier to measure the membrane potential of the cell and control it by passing current into the cell so that the current that we pass is equal to the current flowing across the surface membrane. We can step depolarize the cell, if you will, to simulate an action potential at the top. This produces a rise of calcium similar to a normal systolic calcium one, and this is the current passed by the patch clamp amplifier. Under these conditions, this downward deflection here is the opening of the so-called L-type calcium channels. These channels first open and then they close or inactivate. We wanted to know how much calcium enters the cell. This is current, this is time. I'll hide that, okay. This is time. And if we multiply the two together and integrate it, the shaded area here tells us how much calcium has entered. And on this one beat, somewhere between 3 and 4 micromoles of calcium have entered per litre cell volume. That's small compared to the amount in the sarcoplasmic reticulum, which is perhaps 100 micromoles per litre cell volume, but it's still important in calcium regulation. That's calcium entry. When we repolarize here, calcium is high and the sodium-calcium exchange pumps calcium out. This is the current it generates, which you may be able to see as it relaxes to the baseline. I've amplified it here. This is the integral of the current. And as you can see, the amount of calcium pumped out of a cell here is identical to that goes in. Now, you might say, hang on, there's something funny here. This integral here looks much smaller than that. But remember, when a calcium ion comes into the cell, it brings both its positive charges with it. But because of the 3-to-1 stoichiometry of the exchanger, when it's pumped out, there's only one positive charge. So you have to mentally multiply this by 2. So in the steady state, calcium entry equals calcium exit. So what controls the size of the calcium transient? I want to say now a little bit about calcium buffering. Most of the literature, when people ask the question, what controls the rise of calcium, they think about alterations in all these various calcium fluxes. Now, these calcium fluxes are very important, but they're not the whole story, because in the heart, in the cytoplasm, the vast majority of calcium is not free ionized calcium, but it's bound to buffers. In a typical cardiac myocyte, about 99% of the calcium is bound to buffers, and I guess it's probably similar in most of the cells you work on. So it's like the classical iceberg, only rather than having 7/8 below the sea level, it's 99% below. Well, we can measure, and therefore what that means is that changes of calcium buffering may have as big an effect on the size of the calcium transient as do changes of fluxes. Well, what are the main buffers? There are two. One is Circa. We often think of Circa as a pump, but it binds calcium before it pumps it, so it's an avid buffer. And the other is troponin on the myofilaments. We can measure calcium buffering in intact cells. I'll happily talk about this afterwards. This is the free calcium as measured with an indicator. This is the total calcium that we measure from the sodium-calcium exchange current as it pumps calcium out of the cell. This is the relationship that we get like this. It's a sigmoidal relationship. Look at the scales. This is micromolar or so. This is almost 100 micromolar. If I plot a straight line to the steepest part, it's got a slope of 130. In other words, in this cell, less than 1% of the calcium is free. Most of it is buffered. So one obvious question is, what happens if we change the amount of calcium buffering in the cell? And we did this several years ago by adding niter-5, which is a Bacter-like calcium buffer. This is from a cell in the absence of calcium buffering, or exogenous calcium buffering. We get these size calcium transients. We'd also had in the same cell a lot of niter-5, and you can see that the calcium transients are smaller with added buffering than without it. Well, that won't surprise you. For a given size flux, the greater the buffering, the smaller the rise of free calcium. But as well as an effect on the magnitude of the calcium transient, you can also see, I think, the kinetics. These look slower than those. And that's shown if we normalize the two to the same peak level. With increased buffering, the calcium transients decay more slowly. And the reason for that is that a certain flux here, produced by Circa, in the presence of increased calcium buffering, will produce a smaller and therefore slower decay of calcium. So we would argue then that changes of calcium buffering may be very important in determining not just the amplitude, but also the rate of fall of calcium. And right at the very end of the talk, I'm going to show you this is indeed the case and suggest that this is something that has been overlooked, at least in cardiac muscle. Okay, forgetting about buffering for a moment, what else controls the size of the calcium transient? Well, on this diagram, there are three obvious control points. One is the size of the number of L-type calcium channels that open, because the more that open, the bigger the rise of calcium to open the ryanodine receptor. Another one are the properties of the ryanodine receptor. And finally, the amount of calcium in the sarcoplasmic reticulum. In today's talk, I'm going to concentrate on the amount of calcium in the sarcoplasmic reticulum, what it does and how it's controlled. So I first want to show you that SR calcium is a major regulator of the calcium transient. So here we've got an experiment where we measure SR calcium content. And let me explain, this is expressed in terms of micromoles of calcium in the SR per litre cell volume. So it's not the free concentration in the SR. This is the size of the calcium transient. These days we can make these measurements by trapping fluorescent indicators in the SR, but originally we did it by using caffeine to release calcium into the cytoplasm and work out how much calcium was in the SR before. It doesn't matter which way you do it, you get the same result. And the result is, to nobody's surprise, as you increase SR content, you increase the size of the calcium transient. But you'll see it's not a linear relationship. We can curve fit it, and on average, the amplitude of the calcium transient, the free calcium transient, is proportional to the SR calcium content cubed. So an obvious question is, why is it such a steep relationship? I think there are several factors here. If we increase SR content, then each time a ryanodine receptor opens, more calcium leaves through that ryanodine receptor. Secondly, as we increase SR content, more ryanodine receptors open, as I told you before. That's two factors. The third one, I think, is that as we get to very large amounts of calcium release, we start to saturate the cytoplasmic calcium buffers. So a given extra release of calcium produces a larger increase of free cytoplasmic calcium. Whatever the explanation, we think that this steep relationship is tremendously important for cardiac function. And let me try and explain why. So here's our steep relationship. What it means is that even a small change of SR calcium will have a big effect on the calcium transient amplitude and thence on contraction. So if we increase heart rate, we get an increase of SR content, and that will partly account for the increased force of contraction of the human heart. But it also relates to disease as well. We know that in heart failure, the size of the calcium transient gets smaller, and it's been suggested that this could be due to a decrease of SR content. My colleagues here were interested in that. So they produced a model of heart failure where they put a band around the aorta so the heart has to pump against a greater resistance, and eventually it fails. And when it fails, as many people have shown before, the calcium transient here, the systolic calcium transient, is smaller. On average, it's about half, 0.47 of the control level. So they asked the question, can this be accounted for by a decrease of SR calcium content? So they measured the SR content in control and heart failure. And you can see the SR content has decreased, but the decrease of SR content is only to about 3/4 of control. So the question is, can this modest decrease of SR content account for this bigger fall of systolic calcium? I think I've given you the information already to show it can. I've told you that the calcium transient is proportional to the cube of SR content. 0.75 cubed is 0.42, and 0.42, given all the errors which you can only guess at in these experiments, is pretty similar to 0.47. So don't get hung up on the numbers. All I want to say is that because of the steepness of this relationship, even modest changes of SR calcium content are important in controlling cardiac function. The final reason we're interested in this relationship is to be teleological. It presents the cell with a problem. If the heart wants to have a constant force of contraction from beat to beat, it has to have a constant size of the calcium transient, and that, because of this relationship, means that the SR content must be controlled from beat to beat. And that raises the question of how is SR calcium content controlled in the heart? And that's my next question. If you go to textbooks, you'll see a lot of discussion of the fact that SR content is changed by regulation of SERCA, by phosphorylation of the accessory protein phospholamban, by these two kinases. Now, I've got no doubt that these kinases do phosphorylate phospholamban and therefore thereby affect SERCA, but I don't think this is the major factor responsible for beat-to-beat regulation of SR content. Rather, what we think is important is something we've called autoregulation, whereby calcium released from the SR affects movements of calcium across the surface membrane. And I want to give you some of the evidence for that now. This is a very simple experiment. We're stimulating the cell very slowly, electrically. We then apply caffeine at 10 millimolar. That releases calcium from the SR. Calcium rises to a high level. It doesn't stay there. If you did this experiment in skeletal muscle, it would stay there, but it gets pumped down within a few seconds because this rise of calcium activates the sodium-calcium exchange. Calcium is pumped out of the cell. At this point here, when we wash off caffeine, resting calcium is normal, but because there's no calcium in the SR, when we stimulate, we get a very small calcium transient. The SR is empty. Over the next 20 seconds, however, the calcium transient increases, and that's presumably because the SR is full. So it's occurred to us that if we understood how the SR is recovering here, this might tell us how SR content is controlled under normal conditions. So I'm going to show you a similar experiment now, but I'm going to quantify the underlying calcium fluxes. Identical experiment, we start off with the SR empty, then it becomes full. And I'm going to show you now calcium influx and efflux with the SR full and the SR empty. This is the L-type calcium current when the SR is full. When we depolarize, as I've shown you before, the channels switch on and then close or inactivate. And if you mentally integrate that, that tells you how much calcium enters the cell here. I'm now going to lay on top of it the corresponding record from the same cell when the SR was empty in red. You can see the activation is identical, but the inactivation is much slower, so that the amount of calcium that enters here and here is much greater. And the explanation for that was known from the work of others long before us, which is that this inactivation of the L-type channel is a calcium-dependent process. So the bigger the systolic calcium, the faster the inactivation, and therefore less calcium enters the cell. More calcium, faster inactivation, less calcium entry. That's calcium influx. What about the exit or efflux? That's shown here. This is the sodium-calcium exchange current when the SR is full, big sodium-calcium exchange current, lots of calcium is pumped out. When the SR is empty, as shown here, the current is much smaller, the amplitude of this current. That's even easier to understand. Sodium-calcium exchange is controlled by cytoplasmic calcium. As we increase the size of the systolic calcium transient, more calcium is pumped out of the cell. So we've got two surface membrane fluxes here, the L-type channel and the sodium-calcium exchange, whose activity is controlled by the size of the systolic calcium transit. And we think that's fundamental to understanding the control of SR calcium. I've shown you this diagram several times before. Calcium enters, the ryanodine receptor releases calcium, and we get a calcium transit. The main reason for having a calcium transit is to activate the contractile machinery. But there are other targets for calcium, and I've just told you about two of them. One of them is the L-type channel. Calcium will decrease calcium entry by inactivating it. The other is the exchanger. Calcium will increase calcium efflux by activating it. So imagine if one has an increase of SR content here. I've told you this will increase the size of the calcium transient. Because of this, it will decrease calcium entry and it will increase calcium exit. And the combination of decreased entry and increased exit will decrease first cell and then SR calcium back to control levels. And we think this incredibly simple negative feedback mechanism is how the cell controls its SR content. Changes of SR content change the calcium transit amplitude. These affect movements of calcium across the surface membrane and then feedback to control SR calcium. I want to say two more things about this mechanism. In your community, I'm sure lots of you are interested in store-operated calcium entry. And a major mechanism for store-operated calcium entry is where stim on the endoplasmic reticulum affects calcium entry via ORI on the surface membrane. So if the endoplasmic reticulum calcium decreases, a signal is sent to the surface membrane to allow calcium to enter to refill the endoplasmic reticulum. I think this negative feedback mechanism in the heart achieves exactly the same thing, but it doesn't do it with the classical store-operated mechanisms Rather, it does it with the calcium current and the sodium calcium exchange. Well, you might ask, why doesn't the cardiac myocyte, the ventricular myocyte, have a conventional store-operated mechanism? All I can do is speculate and point out the calcium fluxes across a cardiac membrane per second are much, much greater than those in most electrically non-excitable cells. And it may be energetically much more efficient to regulate these fluxes, which the cell needs in any case, rather than to bolt on an additional store-operated mechanism. But that is just speculation. The other thing I want to say is that when I show this, people sometimes say, well, given this feedback mechanism, doesn't that mean it's impossible for the SR content to change? Well, actually, nothing could be further from the truth. If you look at this feedback mechanism, the precise level reached will depend upon the expression and activity of all the calcium handling proteins, the L channel, the ryanodine receptor, SERCA, and sodium calcium exchange. Change the activity of any of them and you will change the set point. And this is tremendously important, for example, in heart failure. I've already told you that in heart failure, SR calcium content decreases, but I didn't explain why. It comes down to the fact that several things change in heart failure. First of all, in heart failure, you get an increased expression of sodium calcium exchange. So more calcium is pumped out, leaving less in the SR. The ryanodine receptor becomes leaky and starts to open also at diastolic calcium, depleting the SR. And finally, SERCA activity decreases. Each of these alone, and certainly their combination, leads to a decrease of SR calcium content. So in heart failure, this negative feedback mechanism is still working, but the set point is changed because of the change of these expressions. And I think also in most maneuvers that change the force of contraction of the heart, you get changes of the set point of this mechanism. Okay, that's all I want to say about how calcium rises. Now I want to talk about something we've got interested in more recently, which is how calcium, what sets the diastolic level of calcium. And if you're interested, we've reviewed this very recently. So let me remind you about diastolic calcium. Diastolic calcium has to be low enough that the heart is sufficiently relaxed that it can refill with blood from the venous system. Compared to the enormous numbers of papers on the control of systolic calcium, there's virtually nothing known about what sets diastolic calcium. And again, we can talk about that later if you're interested. So it's required for normal cardiac function. We know that in heart failure, many patients with heart failure have an increase of diastolic force and possibly calcium, so their hearts can't refill with blood. And indeed, there's a form of heart failure called heart failure with preserved ejection fraction, where at least at first, there's nothing wrong with the heart's ability to pump blood out. It just can't refill with blood. And this is probably accounts for half the cases of heart failure today. But as I've said, we know very little about the control of diastolic calcium. So we thought it would be interesting to see how the surface membrane and the sarcoplasmic reticulum control diastolic calcium. And I want to present to you today some reasonably recent experiments looking at the effects of sarcoplasmic reticulum on diastolic calcium. So why have we focused on the SR? Well, that's because, as I've told you, in heart failure, the ryanodine receptor becomes leaky and SERCA activity decreases. So we thought it would be interesting to know what happens if we reproduce these in normal cells. So let me emphasize, the experiments I'm going to show you now are done on normal rat ventricular myocytes, no heart failure. We are using pharmacological means to mimic what happens in heart failure so that we can look at processes one at a time rather than the whole panoply that occurs in heart failure. So let's look first at a leaky ryanodine receptor. We stimulate at a slow rate. These are our calcium transients. We now add 2 millimolar caffeine to make the ryanodine receptor leaky. This results in calcium being pumped out of the cell. It's lost from the SR. We reach a new steady state here where there's a large decrease of SR calcium content, but there's still some calcium in the SR. And as many people have shown before, because the SR content is decreased, the calcium transient here is smaller than here. What hadn't been noted before, however, is that the diastolic calcium rises from here to here. So why should making the ryanodine receptor leaky and depleting the SR raise diastolic calcium? Well, our hypothesis was that it comes back to calcium flux balance and the need to recycle calcium. These are expanded versions of calcium transients in baseline and in caffeine. Under these conditions, the amount of calcium pumped out by the exchanger is proportional to this shaded area here. Sodium calcium exchange activity is proportional to calcium. We know, I've already told you, that the efflux is equal to the influx. When the SR is leaky, we know from our patch clamp experiments that influx is not changed. But the systolic efflux here is much smaller than here, and therefore the efflux is less than the influx. Well, that can't persist in the steady state. If it did, the cell would be gaining calcium. So what it looks like the cell has done is to elevate diastolic calcium so that at the diastolic level, the exchanger is pumping calcium out of a cell, so that possibly the sum of diastolic and systolic calcium, now the total efflux, now equals the influx. That's our hypothesis, but all I've done here is to shade in a few traces, as any child could do. Can we quantify it more to see if this is indeed the case? So our hypothesis then is that the rise of diastolic calcium is required to maintain flux balance in the cell. This is a similar experiment, now with only 1 millimolar caffeine, so the effect is slightly more modest. Basal conditions, we stimulate, diastole, systole, stop stimulating, resting calcium. We release calcium from the SR with caffeine. We've still got some calcium in the SR here, but less than control. And you can see again a decrease of systolic and a rise of diastolic calcium. The grey trace below is the same data. The red is this data put through a low-pass filter to give you an idea of what average calcium is in the cytoplasm. What I want you to notice is that average calcium with a leaky ryanodine receptor is essentially identical to that with a normal ryanodine receptor. So, in other words, the rise of diastolic calcium exactly compensates for the fall of systolic calcium to preserve average calcium. Since sodium calcium exchange pumps in proportion over this range to the cytoplasmic calcium concentration, this means that the sodium calcium exchange flux here is identical to here. So the cell was in flux balance to start, it's still in flux balance here. I also said in heart failure, the S-circuit activity decreases. So let's see what happens in a normal cell if we decrease circuit. We can do that with thapsigargin. And you can see the calcium transient gets smaller. It's been known for years. Decreased circuit. This decreases SR calcium content and decreases the size of the calcium transient. It's got here. But you can also see that the diastolic calcium rises. That hadn't been picked up. And again, we think this is just a compensation to maintain flux balance. This decrease of the calcium transient will have decreased systolic efflux. And this has to be compensated for by a rise of diastolic calcium. And this will therefore increase diastolic efflux such that total efflux equals influx. So we think this analysis applies as much to inhibiting circa as making the ryanodine receptor leaky. So I conclude the main part of the talk by saying it suggests there's a novel role for systolic calcium. Obviously, systolic calcium is there to activate contraction, but we think it also protects against a rise of diastolic calcium. And all other things being equal, if you decrease systolic calcium, you will increase diastolic calcium. So to go as far then as to say that diastolic calcium is controlled by systolic calcium. So if anybody's interested in what happens to diastolic calcium in patients with heart failure, I don't think you should think of it as being a separate entity from what's happening to systolic calcium. You have to think in a holistic way of diastolic and systolic calcium interacting with each other. And perhaps the concept of average calcium is a useful one. So if I've got a take-home message, it's that things like flux balance, recycling and buffering are terribly simple concepts, but they've led us to some things we didn't expect. which is the control of SR content, the control of diastolic calcium, and the role of buffering saturation in controlling systolic calcium. I hope I haven't talked for too long. All I want to do now is thank the many people in the lab who've actually done the experiments, and finally a shameless advert for the journal, which I now edit, and if you can send your best work to it tomorrow, I'd be deeply grateful. But thank you for your attention.
David, thank you so much for this overview. It's been really, really interesting, and it opened my eyes to a few things that I hadn't really thought about before, especially in terms of the diastolic controlling the systolic and vice versa. So this is really interesting, and also the last bit of unpublished data. So if everyone could type their questions in their box, and then I will relay them back to David. After coming in, maybe I can ask a quick question of my own. I was wondering, we've discussed about diastolic calcium and that rising in heart failure. Something that hasn't really been discussed is mitochondrial calcium and nuclear calcium. Do you think it's the increase in diastolic calcium in heart failure that might be driving an increase in nuclear calcium as well, or what are your thoughts on that?
I don't know about what. My guess would be that nuclear calcium depends upon the time average of cytoplasmic calcium, so it'll be affected by both diastolic and systolic calcium. I mean, interestingly, in that one flow-3 trace, you could see that nuclear calcium lags behind. So, but I would guess that both are affecting it.
Gene transcription, and how the increases in calcium might be, driving the those kind of factors.
Sure, I think it's affected by both. Mitochondrial calcium, as I'm sure you know, is a real controversy as to whether there are beat-to-beat changes of mitochondrial calcium that accompany the beat-to-beat change of cytoplasmic calcium. Personally, I'm not convinced that there are at normal heart rates, but others would differ. No,
it's definitely something that needs more investigation, and we should be working on that. So first of all, Martin's asked a question, basically saying, not all positive inotropes are affecting myocytes via the same signaling pathway. So indeed, we've got adrenergic stimulation versus endothelin and angiotensin, but they all seem to give sustained increases in systolic calcium. And his question is basically, does that all fit into the scheme that you've been proposing?
Yeah, I mean, that's okay. So on my scheme, a rise of systolic calcium, the simplest way to get it is if you have an increase of calcium entry into the cell, because if the duration of the calcium transit is unaffected, if you get a rise of systolic calcium, that means more calcium is being pumped out of the cell. And I think the only way that can occur in the steady state is indeed by increasing calcium entry into the cell. The complication, of course, comes that a maneuver that stimulates circa, such as beta-adrenergic stimulation, beta agonists, will accelerate the fall of the calcium transient, and that will decrease the calcium being pumped out of the cell. So in that case, you can get a bigger calcium transient, but it's briefer without having an increase of calcium entry into the cell. I hope that makes sense.
Yeah, it does for me. I hope it does for everyone else as well. So I've got another question from Shmuel Mualem, who asked, what is the contribution of phosphate-rich compounds, so for example, ATP, CP, to calcium buffering in the heart?
It's very marginal. I mean, it's very small. I mean, you can ignore it compared to circa and troponin. You've got, it's about 100 micromolar on troponin with its KD, it and circa dominate. That review article, one of the review articles has all the values. It's small.
Okay, great. We have another question from Mohammed Trebek, who basically says, you speculated the coupling of STIM1 to ORAT channels might not be cost effective, and that's why we speculate that this scheme is the case here. So he thinks that ORAI involvement in beat-to-beat refilling of SR is unlikely simply because the store-operated channel pathway is too slow. So it's a speed issue. However, do you see any alternative role of calcium channels, calcium signaling through the ORAT channels in hypertrophy and remodeling? And, is there any issue on transcriptional regulation, for example? So I guess this is getting back to the nucleic.
Indeed, Molkent and Hauser and probably other people I've forgotten have indeed suggested that store-operated mechanisms are very important for some of the possibly adverse remodeling in hypertrophy. The other thing I should say is that there are papers out there suggesting that store-operated channels store-operated behavior can be seen in cardiac myocytes. Most of it has been seen in neonatal cells, but there are some studies claiming it in adult myocytes. It's just that we don't see it.
Do you think that's a species-dependent issue, or what are your thoughts on that? Is it is it being looked at in humans, or?
I can't think of the human literature. We've looked at it in things from mice to sheep, which covers most of the gamut of things I know about.
Okay, so Luke Laybart asks, is there a reason for NCX activity at steady state during diastole then?
Well, just that I think over that period, the whole cardiac cycle, you need to pump calcium out to balance the calcium that's come in. The exchanger, if you look at it, its activity is roughly proportional to calcium. So I don't think it's got a choice other than to pump calcium out in diastole, unless I'm missing something.
So it just has to do because of the concentration gradients, basically.
Yeah, well, just because of the concentration of calcium in the cytoplasm.
Yeah, exactly.
Yeah. I mean, what I don't see any evidence for in neurons, you've got the nice work from years ago from Bourget and DiPolo suggesting that PMCA was there to control calcium at low levels, and NCX controlled calcium when you've got loads on stimulation at higher levels. In the heart, any studies I've seen, anything we've done, suggest they both work over the same range, that you don't get that distinction.
No, indeed. And it also can, because it can be operating in both modes as well, right? So this is also what in terms of what the NCX is doing.
Yeah.
Kevin Foskett also asks, plasma membrane voltage membrane affects the driving force for sodium calcium exchange. So are changes in membrane voltage also associated with heart failure, for example? And is this factored into your thinking about fluxes through the sodium calcium exchanger?
It's factored into our thinking. It's not, unfortunately, factored into our experiments because we do them under voltage clamp, where, for simplicity, where it stays, the duration of the voltage clamp is the same. You get a modest prolongation of the action potential early on in heart failure, which will decrease the ability of sodium calcium exchange to pump calcium out of the cell. It's an added complication, but I don't think it would change the overall take-home message.
Okay, so it's just the actual difference it will make are probably pretty minimal to the main issue that's afoot.
Well, I still think that calcium entry has to balance calcium efflux. If the change of the voltage profile of the action potential makes it harder for calcium to be pumped out, that will mean you have to get a bigger average calcium, which could be due to a bit elevated diastolic or systolic calcium.
Okay, so in the end,
other people have modelled it.
Yeah, in the end, it needs to balance out, basically. That's what it boils down to.
Yeah.
So Antonio Villalobos asks, how do calmodulin mutants affect the control of L-type calcium channels, affecting the steady-state calcium fluxes?
We haven't looked at that. I mean, they affect calcium inactivation, and they also of the calcium current, and they also affect control of circa, but I haven't looked in any detail, I'm afraid, at overall balance.
So this was something I was actually going to jump on a little bit. The unpublished data you showed with the rate dependence. Have you looked at that with CAM kinase inhibitors, for example? Could this be the calmodulin kinase also acting as a buffer? Or it'd be interesting to know what role CAM kinase is playing there, because obviously we know calcium causes a CAM kinase increase phosphorylation, but is it having other effects as well?
We haven't done that, no. The values of the concentrations of calmodulin doesn't suggest that it's a big buffer compared to the other ones, but that's a poor substitute for actually doing the experiment, I agree. Okay,
no, it was just something that came to mind. Sure. So Don Gill asks, are you saying there are rapid mechanisms for changing cytoplasmic buffering? And if so, what are these mechanisms?
No, I'm not. Well, that's interesting. So what we've looked at is phosphorylation. So circa, if you phosphorylate phospholamban, circa has a higher affinity, and I think you'd expect that therefore to give you a higher affinity buffer. If you phosphorylate troponin, it gets a lower affinity, and it might give you a lower affinity buffer. we've seen these in cells, these effects. I haven't measured the kinetics of it, but they seem to occur quite quickly. No, I mean, that's a good point. I think I'm more thinking about, well, what I showed at the end, which is changing diastolic calcium effectively changes the buffering power. But it's perfectly possible in heart failure, for example, you may get changes of calcium buffering due to changes of circuit that are known to happen. And I don't think this has been factored in.
And we also know, for example, like the HRC, so the histidine-rich complex can also buffer higher when it's lower, when calcium levels are higher, and more when it's lower, right? So there's also a seesaw effect, right? As far as I understand. So that could be another way of buffering, I guess, right?
Yeah, although I mean, the lower affinity buffers will buffer at higher levels. I mean, most buffers buffer more strongly at lower calcium levels than at higher, with the interesting exception of calsequestrin, which varies, the SR one, which buffers more strongly at higher calcium.
Okay. So Agnieszka Jerda asks, sorry if I got the pronunciation wrong, asks, is there any role for pannexins or connexins in heart calcium homeostasis? And Luke says exactly.
I'm going to defer to Luke because, you know, he's the man who knows about this. We've corresponded. I'd love to know more about it. But Luke, tell us.
Wow. Can you hear me?
Yeah, perfectly. Yeah. Thank you. I
will refrain of saying anything, but maybe that would be a good idea for next webinar. So I can for an hour or at least half an hour, but that would be a nice idea. So yes, we think, so there's a paper in the pipeline where we have quite some evidence that hammer channels contribute to calcium entry and especially also to extrasystoles and DADs, and so yeah, there's quite some momentum for thinking about the role for pannexins. Pannexins in our hands in ventricular cardiomyocytes, we didn't find evidence, at least not under diastolic conditions. So what I'm talking about is diastolic conditions show that if you apply caffeine, you open heavy channels with calcium and caffeine, so chemical effects not linked to the voltages at minus 70 or minus 80, and that contributes to DAD generation. So, but that is for another another session, I would say.
You've got a volunteer, Dan.
There we go. Yeah, no, thank you. No, that makes complete sense. We'll speak to Martin and put you guys down for a next webinar. So where were we? Yes, that one. So Pepekin asks, what is the contribution of high affinity binding sites on SR calcium buffering protein to systolic buffering, particularly when the RYRs are open?
You mean either way, intra-SR calcium buffer sites or extra-SR? Intra-SR?
They're just saying SR calcium buffering protein. I'm imagining he means intracellular buffering. Or maybe you can come back with that and let us know. So actually, there's a question coming back from what Luke and you were saying previously. Something we haven't really touched about is the location, right? And these microdomains of calcium. And, you know, obviously we have the connexins, which are going to be on the end of the cell, you know, at the intercalated disc, whereas we have our T-tubules where we have probably a much higher microdomain calcium, right? Do you think that you might have a microdomain of calcium at the intercalated disc as well then, or do you think that might be the case?
Well, that's very interesting because there's very little SR at the intercalated disc. Mm-hmm. So although calcium will move from one cell to another, in our hands at least, calcium waves don't propagate from longitudinally from one cell to the next. So we do it with great difficulty. And I think that's because there's little SR at the ends of the cell. I mean, the other thing that we're interested in, which I know nothing about, in terms of calcium buffering, is at those release sites, there's probably a lot of circa but there may be very little myofilament, so there may not be much troponin buffering it. So,
yeah, no, indeed, this is something that's, because people talk about coupled and non-coupled randin receptors and where they are in terms of with the T-tubules, but no one really discusses coupled and non-coupled circa, right? Because we don't, it's very difficult to image and we don't really know what's going on there. So I think there's definitely a lot, you know, we still should be in a job for a while, I think. Which is not a bad thing. So Martin de Smet asks, many pharmacological compounds have been shown to modulate systolic calcium transients and are proposed to be used in heart failure with reduced ejection fraction. So for example, beta blockers, NCX blockade. In heart failure with HFpEF, so with preserved ejection fraction, it seems that these agents aren't that effective. So do you think this is down to differences in intracellular calcium handling, or do you think this is more related to the diastolic calcium handling?
well, first of all, I'm not sure how effective drugs that work on systolic calcium are even in conventional heart failure with reduced ejection fraction. I mean, there's a pretty dismal story, I think, about most positive inotropic agents that work by increasing calcium. I mean, somebody once said it's a bit like trying to flog a dead horse. You don't make it walk any faster. I mean, I think it's fascinating to know how you might differentially treat diastolic compared to systolic calcium. We speculated a bit about that, but I think we just need more information.
Yeah, and there's some recent work from the Goldhaber lab, right, that was looking at how the HFpEF myocytes are changed and the calcium handling. It's really interesting data coming out. So I think this is definitely something that needs to be looked at more, because we don't really know that much about the diastolic function about it, which is why I really liked your idea of the diastolic driving the systolic and vice versa. It's a really interesting idea. Okay. Is there anybody else with any questions? We're nearly at the end of our hour. So if there's no more questions, then it just leaves me to thank all the participants, and also obviously thank you, David, for a really, really interesting presentation and the discussion afterwards. And also thanks to Katja and Martin for organising this session. It's been very enjoyable. So thank you.